Hi, everybody, and welcome to the fifth webinar of BioMeca. Today, we are going to speak about how defining new types of markers in the skin with atomic force microscopy. So, I am Céline Paillier. But more than introducing myself, I wish to talk about how characterizing skin properties is crucial to understand its mechanisms and to develop the best dermocosmetics products to enhance skincare. Briefly, BioMeca is specialized in biophysics and life science with a focus in skin biology. Our innovation team is dedicated to provide tomorrow's technologies for your research. During this webinar, I'm going to talk about the current type of markers used in skin biology and a new one, the mechanical signatures or mechanomarkers. I will move on to a very brief explanation about AFM and how this fantastic technology can bring us insights about mechanical properties of skin and allows us to define mechanomarkers. Finally, we're going to see which mechanical signatures could become markers of skin conditions and a conclusion with perspective. So there's a lot of biomarkers for skin disorders. Here we will define skin markers related to intrinsic factors as aging, but also to external stress that could lead to inflammation and diseases as dermatitis. Biological markers have been defined as cellular, biochemical, or molecular alterations that are measurable in biological media, such as human tissues, cells, or fluids. In practice, biomarkers include tools and technologies that can aid in understanding the prediction, cause, diagnosis, progression, or regression of health conditions. Skin markers measurements have enormous value for detailed assessment of skin conditions, both for clinical applications and skincare. However, there exist different reasons why most potential biomarkers failed in achieving adequate sensitivity and specificity and are not accepted as clinical tests. One main reason is that most biomarkers are dealing with detecting diseases at an early stage in humans with different sex, age, ethnicity. And other important fact is to find a protein or a metabolite on an extremely low concentration level is very difficult. To improve it, there are different strategies, and the potential solution should be to improve the assay, combine several markers, or stratify population. Here, BioMeca suggests to upgrade the list of biological markers with mechanical signatures given by AFM measurements. Current markers encompass a wide range of biological material that can be used, such as morphological information with immunofluorescent or immunohistochemical staining, and filaggrin, for example, to detect atopic dermatitis, genomic information with P53, 16, 21, transcriptomic profiles obtained by analysis mRNA, or TGF-beta to detect healing, for example, and proteins such as interleukins or metalloproteases to detect senescent cells. Here an example of what we did at BioMeca with those different markers. And for example, to prove aging alters the production of extracellular matrix components by fibroblasts. As we can see, aged fibroblasts produce less fibronectin and collagen 1. And by stimulating these aged fibroblasts with an active ingredient or TGF-beta in this example, we can restore the young phenotype. Now I will talk about how AFM helps to define those new markers about skin conditions. Here a quick reminder of the AFM. The principle is based on indentation exerted by a cantilever with a spring constant on which we can apply a force that allows the movement of the cantilever. To process the signal, a laser beam hits the cantilever to reflect on a photodiode to 4 quarters, and we record the shift of the laser beam on the photodiode. The raw data obtained is a force indentation curve. Here, the different stages of the curve. The first one with a flat curve corresponds to the approach stage. Then we have the exponential curve, which is the indentation. And finally, in red, the retract curve. We have two main modes with AFM. The contact mode, where the tip is all the time in contact with the sample. The force applied remains constant because of a piezomotor controlling the height and adapting its position. With this mode, we obtain topographical data. The second mode is the quantitative nanomechanical mode or tapping mode. The cantilever hits punctually, oscillates on the sample. This QNM mode provides live force tomography that I will explain later. So now we have reviewed the current markers used in biology and the AFM technology. We are ready to talk about this new kind of markers, the mechanomarkers defined at BioMeca. I'm going to begin from the top of the skin with corneocytes. These cells are essential in the skin barrier function as they form a cohesive layer which provides a water-resistant barrier at the skin surface. We carried out a clinical study with L'Oréal on 20 children with atopic dermatitis and 7 healthy children. Corneocytes have been collected thanks to tape stripping, a complete non-invasive method allowing the removal of the horny layer. The objective was to understand the maturation process of corneocytes and how atopic dermatitis, named AD, affects skin integrity. On this slide, we show topographical images of healthy corneocytes and a corneocyte from atopic dermatitis sample. We can easily see a characteristic between them. On the right, after zooming, we see that the corneocytes on the atopic dermatitis sample got a lot of asperities, whereas the healthy one is smoother. Then we went further in the understanding of the mechanical influence of AD on corneocytes. We carried out AFM measurements to assess corneocyte stiffness. Filaggrin is commonly used as a marker for AD, but mutation in filaggrin gene is not sufficient to induce AD, as some people positive for the mutation does not present the pathology, and conversely, some AD patients do not possess the mutation in filaggrin gene. We assess corneocyte collatin on AD patients with filaggrin mutation and wild type, and same with healthy patients. As you can see on the graph, AD patients had significantly lower elastic moduli of corneocytes compared to healthy controls. In conclusion, thanks to AFM, we highlighted the fact that mechanical markers can be used for AD diagnosis. In addition, we have defined two new markers, a morphological one and a mechanics one. Following this study, we realized the potential of these mechanomarkers to diagnose skin disorders and also the importance of the biological samples collection. So we have been developing a device including these two characteristics: the harvest of the first layers of the stratum corneum via a completely non-invasive system, and using the AFM system to characterize the AD grade from mechanical stage of the corneocytes. Here, we suggest to use corneocyte stiffness as an aging marker. We perform high-resolutive image and AFM measurements on skin sections, and we focus on two characteristics: topography of the stratum corneum and stiffness mapping. From this data, we can extract and follow the evolution of the skin barrier properties over time in measuring the number of the stratum corneum layers, their thickness, and stiffness, as we know that stratum corneum thins and stiffs with aging. Then, to specifically evaluate stiffness evolution during aging, we harvested by tape stripping, again, corneocytes from two age groups, 20-50 and 50-16 years old. Then we performed AFM measurements and concluded that stiffness of corneocytes increases twofold between 2060 and 50, 60 years old. Thanks to this new mechanical insight that aging impacts the stratum corneum, we decided to go further in the understanding of the epidermal cell renewal. We focus on cells of basal layer that are anchored to the dermal epidermal junction. This junction exhibits undulations, red ridges, and dermal papilla, which vary with aging. Indeed, the EG flattens, phenomenon concomitantly observed with a decrease of epidermis, renewal, and desquamation. Thanks to MCASP and call for markers, we localize some of these basal cells to measure their mechanical properties. Here, we demonstrated that basal layer cells have specific mechanical properties depending on their localization on the DEG. Indeed, we performed some stiffness map along the DEG and revealed that cells located above dermal papilla have higher cellular stiffness than red rigid cells at all ages. This indicates that specific cell mechanical properties exist along the DEG in epidermal basal cells. Then we wanted to better elucidate the DEG mechanical evolution over time. Here you see a 3D force map of dermal epidermal junction with atomic force microscopy. Each pixel corresponds to a stiffness value where red is low stiffness and red a high stiffness. Green indicates a low stiffness. We demonstrated that the stiffness of the DEG increases with aging, and this represents a new solution to assess skin aging, focusing on DEG. Now I will talk about live force tomography. This is an analysis process to reconstruct in 3D the skin mechanical behavior thanks to a dedicated algorithm developed by BioMeca. LFT allows us to provide a 3D representation of biochemical characteristics of tissues in quasi-physiological conditions. Then, we were able to follow the evolution of the elastic modulus in dermis and use it as a mechanical marker. At the cellular level, LFT can be used to follow another mechanomarker, as for example, actin network. We carried out AFM measurements on melanocytes, treated or not with an active supposed to stimulate actin filaments polymerization. Thanks to LFT, we demonstrated that this actin ingredient induces subcellular stiffness increase, meaning an actin network stabilization. We did more, and we improved resolution of the measure to understand the physical impact at the nanomolecular level. We were able to show a contraction of the actin cortical network. This innovative approach can be used to monitor the effects of ingredients or stimulus factor on cell or tissue response. BioMeca's innovation team has been working hard for 4 years now, and we are ready to talk about this second device, including our know-how in mechanical properties characterization of skin. This device gives you in real time the evolution of skin mechanomarkers. Easy to use, connected to the cloud, process data could be directly used for clinical studies. Now, time for the conclusion. What's our perspective for research and skin diseases diagnosis with this mechanomarker? To conclude, I would say that atomic force microscopy, both as an imaging and as a mechanical tool, provides an excellent opportunity to study soft biological samples such as skin. AFM becomes a multifunctional toolbox to observe and measure various biophysical parameters of cells and tissues. AFM can be used in physiological conditions and also does not require elaborated or specific sample preparation. Skin biology is complex, but mechanical characterization of tissues is an amazing tool to predict and diagnose skin disorders. AFM and mechanomarkers represent then a new way to detect any skin disorders, leading to a modification of the skin structure of thickness. Our innovation team at BioMeca is dedicated to seek out solutions to help understanding this complex tissue and also provides new tools to enhance skincare. I hope you found this webinar informative with new insights about how expertise and know-how. So thank you very much. Have a good day.
Thank you. Bye, everybody.